So November is the month of Alzheimer's disease awareness, and I'm glad to today introduce our collaborator from CHU Montpellier, Florine Leib. She is our PhD student working since the last 3 years together with us in the research group of Professor Christoph Hirz. And I'm glad to introduce her today to talk about our research that she's currently performing in the laboratory. Florine, the stage is yours.
Hello everyone, and thank you for being here. So today I'm going to present part of my PhD work, which focused on developing a targeted LC-MS assay for the quantification of Alzheimer's disease blood-based biomarkers. So first, I'm going to introduce the Alzheimer's disease pathology and the technology I used for this work. Then I'm going to explain the objectives we had during this research, explain the method I developed and the assay with development, analytical, and clinical validation, and then expose the outlooks of this work. So first with the introduction. So what is exactly Alzheimer's disease? So Alzheimer's disease is a global health issue, which 55 million people are living with dementia worldwide, and the cases are rising and will triple by the end of 2050. AD accounts for 60 to 70% of all dementia cases, which makes it the leading cause of dementia worldwide. And the age onset is between 70 and 75 years old, with 2/3 of the patient being women. And it's also a huge financial burden for society, including for medical care and social care, which makes it a global health issue. So what are the clinical symptoms? So first, Alzheimer's disease leads to cognitive impairment with memory, language, orientation, and visuospatial perception impairment. Then it can also impact the functional properties of the person with daily life activities, impairment, mobility, and balance issue. And it can also lead to psychological disturbance with illusion, depression, anxiety, and sleep disturbances, which can eventually lead to behavioral changes with isolation and aggressiveness in some cases. So normal aging comes with cognitive function decrease, but in the case of Alzheimer's, the decline is way steeper and more severe. So what actually occurs in the brain? So in an healthy brain, functional synapses ensure proper brain cell interaction. The blood-brain barrier is healthy, which ensures proper nutrient supply and waste clearance of the brain. And all cells are presenting structural integrity. However, for when Alzheimer's disease starts, neurotoxic aggregates starts to form, including amyloid plaques, forms of amyloid beta peptides, or neurofibrillary tangles, which accumulate within the nervous system and are composed of phosphorylated abnormal protein tau. Also, this neurotoxic aggregates leads to the activation of reactive cells, including microglia and astrocytes, which together release inflammatory molecules, which sustains chronic neuroinflammation. This neuroinflammation will subsequently lead to neurodegeneration and brain atrophy. Together, this vicious chronic deleterious circle will lead to impaired blood-brain barrier, which will lead to the release of structural protein, which comes from the dead cells, such as GFAP-tau or NFL, for instance. And Alzheimer's disease is impacted by risk and resilience factor, which can either make it more severe or improve the prognosis of the patient. So among the risk factors, we have depression, anxiety, and mental health issues. We also have genetic risk factors, including ApoE4 alleles, presence and family history of Alzheimer's disease. Also, additional metabolic comorbidities, including cardiovascular diseases, diabetes, or hypertension, are also risk factors for Alzheimer's disease. Obesity, sedentarity as well, and also bad habits, including alcohol consumption, tabagism, or bad diet also. But on the other hand, some lifestyle and environmental factors can improve the life of the people and delay the onset of the symptoms, including having an active social life with regular interaction with other people, high education, constant learning, and engagement in creative activities, sufficient sleep, the wear of security equipment, and correction of sensory deficits, healthy balanced diets, sport, and physical activity as well. But now, how it is diagnosed. So Alzheimer's disease is currently diagnosed first with a clinical examination of a neurologist, which established family history and led cognitive testing. And if dementia is suspected, and especially if AD dementia is suspected, complementary testing are led. So first, there is two different types of biomarkers. So we have neuroimaging biomarkers, including PET scans of amyloid PET scans, tau PET scans, or MRI, which can localize amyloid deposition and tau tangle location, but also evidence the presence of a cerebral atrophy. However, these biomarkers and these types of imaging are pretty expensive, and they are not widely implemented in hospitals. And biological testing through biofluid measurements are a nice alternative to neuroimaging. Currently, there is several biofluid biomarkers which are based upon the ITN classification, with amyloid pathology A, tau pathology T, and non-specific AD changing, including neurodegeneration and inflammation, as N and I. So among the core biomarkers, we have amyloid beta peptides, but also PETA species, with PETA 17 being the most powerful and performing biomarkers at the moment. And also we have total tau and NFL for neurodegeneration. But there is a need for tau pathology, which is not linked to amyloid pathology, and therefore new biomarkers have been suggested, including GFAP for neuroinflammation and reactive gliosis, and specific tau fragments for tau pathology, which is not linked to amyloid pathology. So there is several assays that are currently IVD and CFA certified, with one plasma PTO 181 and PTO 217, which have been validated this year. So now plasma is entered into routine testing, and they all rely on immunoassay. And there is then also all the assays that are only for research use, and that includes emerging biomarkers, large-scale panels, and other plasma assays. So what are the current needs? So several studies revealed that there is inconsistent profiles with a patient which does not typically follow the amyloid cascade, with either worse clinical symptoms and which will suggest neurodegenerative co-pathology, or resilience with delayed cognitive decline in patients who have severe biological state but pretty good clinical cognitive staging. Also, other studies revealed that there is multiple subtypes of AD that exist, which can explain the differences of onset, prognosis, progression, and treatment response of AD patients. So there is a need for new biomarker to precise the picture of each patient. Pure AD is the exception and not the rule, as post-mortem autopsy studies revealed, with more than 70% of people having mixed dementia, including synucleinopathies, TDP-43 or tauopathies, and vascular dementia. Also, AD is a complex pathology with multiple subtypes, with over 5 molecular subtypes that have been currently identified, and that can lead to different prognosis. And this can explain the response to treatment, the fact that the response to treatment is inconsistent. Improvement is only observed in 27% of the cases for treatment with lecanemab, which has recently been approved. And this is very variable. And there is notable severe side effects, including bleeding in the brain in more than 15% of the treated patients, which can also show that some patients have different pathways than others, and it's really important to monitor. So we need a personalized care strategy, which takes each patient biological AD profile into account, including environmental and lifestyle factor, metabolic comorbidities, presence of AD pathology, AD molecular subtypes, and neurodegenerative co-pathologies together to bring the full picture. And complete diagnosis, anticipate prognosis, monitor disease evolution, and tailor therapeutic intervention. So the biomarker implementation timeline is a pretty long journey, which starts with discovery in large case studies, then analytical validation following pretty strict guidelines, then a clinical validation to evaluate the clinical relevance of this biomarker. And this is something that is led in clinical research laboratories. And once this is achieved, we can go to regulatory approval and clinical implementation in routine laboratories. But only few candidate biomarkers manage to reach the clinical implementation. Now I'm going to explain the principle of LC-MRM. So targeted bottom-up proteomics starts with sampling. Once the sample is collected, we undergo sample preparation. So the sample preparation in bottom-up proteomics is separated in different parts. So we have first extraction of the proteins, denaturation, digestion with an enzyme to cut into smaller peptides, purification, and fractionation, additional steps if additional specificity is needed. Then once the sample is prepared, it's injected into the liquid chromatography system. Here, the peptides are separated according to their hydrophobicity in reversed phase mode, for example, and then they go to the mass spectrometer. Once they enter the mass spectrometer, they are separated based on their M/Z ratio. So they are ionized, first selected in the first quadruple. If the ion is not the selected M/Z, it collapses. Otherwise, it goes through the quadruple and reach the collision chamber, which is the collision cell, which is filled with an inert gas, and there the ion undergo fragmentation. So for peptides, there is a typical fragmentation pattern with Y and B ions, and then specific fragment ion are also selected and transferred to the detector. So the more ion reach the detector, the more intense the signal is. And then acquisition and data processing. So what are the main clinical interests of mass spectrometry? So first, mass spectrometry targeted assay does not rely on using antibodies, so which is an asset compared to current immunoassay. It can also be used for discovery and routine analysis. It's a pretty versatile and flexible tool where targets can be added, removed, and adapted to any biological question on any matrix. It can also be highly multiplexed and is a gold standard method which ensures absolute quantification and gives structural information, including either a post-translational modification and sequence information. So then we'll go to the research objective of this work. So I implemented two complementary strategies. First, a multiplex LC-MRM panel, which relies on secondary hallmarks of Alzheimer's disease, which is antibody-free, simple, versatile, and wants to be high throughput and ready for clinic. And on the second part, I developed a specific iPLC MRM relying on immunocapture for specific PTM and specific protein targeting. But today I'm only going to discuss about the first part of this project. So the development of the method started with selection of the targets, which encompasses several different pathways, including synaptic function, lipid transport, genetic risk factors with apolipoprotein isoform phenotyping, inflammation, brain cell structure, and systemic homeostasis to try to dig in and discover new biomarkers of specific pathways. So then once the proteins are selected, we go to the proteotypic peptide selection. to be detected sufficiently in mass spectrometry, peptides need to be tryptic, so digestible with trypsin, unique to the protein we want to analyze. The length should be suitable to MS M over Z ranges. And it should avoid PTMs and reactive residues to avoid any unwanted reactivity and variability. So this is the list of peptides I've chose for the 24 proteins I selected. And then the standard selection is something really important also when we want to perform accurate quantitative mass spectrometry. So the gold standard is to use a full-length protein, stable isotope labeled protein, but it's pretty expensive. Another alternative, which is widely used, is to use just the peptides, so the stable isotope labeled peptides that are added at the end of the sample preparation and therefore does not correct for the digestion. For me, I decided to go for something in between, which is a winged stable isotope labeled peptides, which has a cleavable tag, and then can correct also for digestion and mimic the full length protein. So, and are used peptides from GPT peptides company. So then I optimized the MS settings with selection of 1 quantifier and 2 quantifier ions for each peptide. Then the voltages of these specific transitions were optimized, including collision energy and Q1 and Q3 pre-bias voltages, with some big improvements for most of the proteins, as you can see with the mean fold changes observed. And this is adapted for each peptide. Some other settings are consensual choice-based, which is the case for source, gas flow, and temperatures, and that have been optimized as a consensual choice. And these are the specific parameters I've used. Then once the MS method is developed, we can go to the LC optimization. So first, I built a gradient to ensure sufficient and great separation of all the analytes throughout the gradient without making it too long. Then I tested several different columns with several different phases, which included metal-free, bioinert, and different types of silica with different particle size. And the Clariceptor Shimpact column, metal-free and bioinert, was the best choice with the thinner peaks and the best shape overall. Then I tried different types of mobile phases, especially acetic acid was used compared to the regular and more typical formic acid, and gave a very impressive improvement, especially for very hydrophilic peptides, which elutes prior 7 minutes. So we had improved signal for all peptides with twofold median improvements in general. Then I also optimized the flow rate to ensure we have thin peaks, and using a 400 microliter per minute flow rate increased sensitivity, as you can see, for example, for these two peptides. And it did not impair the separation. Then once everything is optimized, we can also optimize the sample preparation to try to detect as much peptides as we can. So this is a pretty simple workflow that was designed to be high throughput and easy to implement in a clinical lab. So we start with 10 microliter of plasma, which is then reduced and isolated, digested. Then the peptides undergo solid phase extraction to remove any interfering compounds or some undigested enzyme. And then it's dried under vacuum and injected. So it's a pretty simple workflow to try to catch as much of the protein diversity as possible, but it also comes with its drawbacks. So this sample workflow was optimized. The enzyme quantity was divided by 2. Different buffers were tested, different types of digestion, and for the solid phase extractions, different types of acid, different types of sorbents, and it was also automated. And this is the final workflow. Simple, automatable, versatile, and high throughput. But it's not suitable for traces or PTM targeted analysis because it doesn't reach the sensitivity required due to huge matrix effect. Then we go to the analytical validation. So the main challenges of quantitation in proteomics is that for our cases, the analytes are endogenously present. There is no blank matrix and a lack of consensual guidelines compared to small molecules. And there is a wide diversity of plasma proteins with different and extreme dynamic range of concentration and a huge matrix effect, and which can induce variability. Then the strategy we selected is to use reverse calibration, where the curve is built in pooled plasma with the endogenous signal serves as internal standard, and the analyte is the stable isotope labeled peptide that we spike at different concentration. And in this sample, it's the opposite. And we use a fit-for-purpose validation with enlarged criteria and which merge CP2 assay guidance and ICH M10 guideline to reach our purpose. So then I selected the calibration ranges for each peptide, with the black points being the endogenous concentration and the line stated for the selected range. The dotted peptides are the ones that I didn't detect it endogenously, and there was 7 undetected peptides, including interesting ones such as GFAP, Tau, S100B, or interleukin 6, but which are really hard to get and usually use immunoprecipitation to reach these proteins. So I conducted several tests, such as linearity and sensitivity, intra-batch repeatability, operator reproducibility, stability, recovery, and carryover. So for linearity, I built 10 to 12 calibration points for each peptide, and the criteria was to be over with an R-square superior of 0.95, which was achieved for most peptides. For sensitivity, only the 7 peptides that were below the LOQ and the other ones were detected pretty easily. Then for repeatability, I conducted 3 different runs with replicates of 2 QC levels. And here you see that for these 2 peptides, some replicates were outside of the limits, but overall the global accuracy was great. For reproducibility, except for 1 peptide, the other operator had good CVs and proper accuracies, which is interesting because it means that this assay can be performed by anyone and in any lab. For matrix effect, so I prepared 3 different samples, one in buffer, one where the spiked peptides were digested outside and spiked onto the extracted matrix, and then a fully extracted sample. And it leads to this results. So you can see in the bottom that the matrix effect was pretty tremendous with over 70%, which is normal because we had all the proteins in plasma digested and no specific preparation. Otherwise, the extraction was pretty good, as you could see in the top with good extraction leads, and this leads to pretty small global recovery, which was enough for the proteins we wanted, but that can also explain why some proteins were not detected. So a good extraction recovery overall, very high suppressive matrix effect, a low global recovery, and low or no detection of low abundance protein, but robust and conservative sample preparation. For stability, in green we have the the ratio, which is corrected with the internal standard, and you see that the ratio is pretty stable over 5 days, even though the endogenous signal drops. So no targets were not detected after 5 days, which is great. Means that the plate can stay in the autosampler for up to 5 days, and the internal standard is an effective correction. So overall, the performances of the method were the following. So 17 peptides were detected and validated in green. And they are suitable for clinical research. It's a quantitative method, repeatable, robust, and stable. And then I performed the clinical validation. So on the add-on cohort. So this cohort was built on patients with memory complaints that come to memory clinic of Montpellier, Nîmes, and Perpignan. Here they undergo several different tests with clinical examination with a neurologist and biological testing. And with this, we had the diagnosis, including the ATN profile and the pathology suspected. So the cohort demographic was the following. So I classified them with their ATN profile, which was built on CSF measurements. It's a real-life cohort, so you can see that AD is overrepresented, which is comparable to what I explained earlier, with AD being 60% of the dementia cases. So this was my analytical design. So there was 414 patients in total, and they were divided in 9 batches. Each batches contained quality control samples and a calibration curve. And overall, as you can see on the right side, the performance of the method were pretty robust over the 9 batches for each peptide, with good CVs and great accuracies. So regarding the main significant results, so I performed several statistical testing. So first, when we compared DAT and continuum, we could see that 5 types of proteins were significant, especially between controls and severe AD, and between the neurodegeneration positive and neurodegenerative negative patients, including PARC7, clusterin, vitamin D binding protein, osteopontin, and the ApoE isoform, especially E4 and E3. So we will focus on PARC7, which was the most interesting results. So as you can see, it was decreased as the disease progresses, and it's following the disease worsening. Other results were comparison were led on the more specific disease diagnosis. Otherwise, however, it's difficult to draw a statistical conclusion because the group size were too small compared to AD. For the specific other diseases, there were like 10 to 20 patients maximum compared to the 200s of AD. So the statistical power is relatively low, but we can see tendencies. So clusterin is elevated also in Parkinson's and reduced in AD. Chromogranin A seems to be elevated in vascular dementia. Gelsolin also seems to be elevated in frontotemporal dementia. Osteopontin also seems to be elevated in mixed dementia. And then I also performed an inter-platform correlation, as some of the targets were also included in the NULAISA neurology panel. For PARC7, the correlation was pretty low, but it was still positive. However, for ApoE, interestingly, the correlation was negative, which can be explained by the fact that the NULAISA has a different targets a different type of the protein, and maybe is measuring something else. regarding ApoE4, correlations were pretty good between the Lumipulse, the Roche, Elexis, and the Neulisa. Then I focused more especially on the ApoE phenotypes because this method enabled to do a MS-based phenotype, which is not yet clinically implemented. So with a barcode, we were able to, in absence prints and subspecific peptides, we were able to establish the full phenotype of each patient of the cohort. All but one patient were correctly phenotyped with this method, which is pretty impressive. So among the major results, we have seen that phenotypes influences the level of ApoE levels. So E4 homozygotes have very low amounts of ApoE compared to the E2, which are the ones that are supposed to be protective. And also effects on the disease severity. The disease is more severe in patients with the E4, E3, E4 phenotype. So the conclusion of this work is that the MS-based phenotyping was achieved, and it revealed the role of ApoE and its ApoE levels and isoforms in the disease, which can serve as a prognosis and therapeutic strategy choice. It evidenced some interesting biomarkers associated with oxidative stress and inflammation or clearance pathways such as PARC7 and clustering, which can help as monitoring biomarker and also being new therapeutic targets. But this biomarker was not performing enough to replace the current diagnosis biomarkers, including p-tau217. And also we need another cohort to confirm and extend the results, especially the differential diagnosis results on different pathology, because in this one, the group number were too small. But it also revealed that MS is a powerful research tool which brings robust and accurate measurement of hundreds of samples for several different hundreds of targets. It's a pretty versatile tool, and it's applicable to various research questions and contexts. So now the perspective and outlooks of this work within the Alzheimer's disease research is that we don't have a need for new early diagnosis biomarkers as p-tau biomarkers are performing very well. Otherwise, we need a biomarker for stratification, differential diagnosis, co-pathology assessment, and monitoring. We also need for wide, well-described cohorts with a lot of patients, longitudinal cohort, different pathologies, to better unveil the complexity of neurodegenerative dementia etiologies. But also the fact that target MS is a powerful tool for research, and it's complementary to classical immunoassays that are used in routine. Thank you for your attention. And I also want to thank my lab and Shimatsu for their participation in this work.
Thank you very much, Florine, for this great presentation. It's lovely to see all the outcomes from the last 3 years. So thank you. And yeah, if there are any questions, it can be texted somewhere in the notes.